In this video, we'll be discussing the creation of the different vaccines and review the performance data of several that have been released or are about to be released. The primary target for nearly all vaccines is the spike protein because entry of the virus into human cells is facilitated by binding the spike protein to the ACE2 receptors. Antibody directed against the binding region of the S protein blocks binding to human cells, and then the virus is unable to enter these cells and grow. All vaccines express the full-length spike protein. An alternative target being used by a minority of vaccine developers is the membrane protein. Now let's discuss the four major classes of vaccines presently under production. They are 1. Nucleic acid vaccines, 2. Viral vector vaccines, 3. Synthetic protein vaccines, and 4. Killed intact virus vaccines. A fifth form of vaccine, live attenuated or weakened viral vaccines, are under development in India but have not been released for use. Let's begin with the nucleic acid vaccines, because the first two vaccines released fall into this category. Both the Pfizer BioNTech and Moderna vaccines are messenger RNA vaccines. They consist of the full-length RNA message of the spike protein that is packaged in a protective lipid nanoparticle. Nanoparticles are injected intramuscularly, and then the lipid nanoparticles are taken up by immune processing cells, dendritic cells, and monocytes. Once the RNA enters the cytoplasm of these cells, the message is used to synthesize the spike protein that then migrates to the membrane surface of cells, where B cells are signaled to produce antibodies against the protein, and T lymphocytes are induced to generate cell-mediated killer T cells that destroy the virus. The Pfizer BioNTech vaccine is called BNT162b2, a randomized clinical trial, RCT, consisting of 43,000 subjects compared 30 micrograms of vaccine to control subjects injected with saline. Of the half who received the vaccine, 8 developed COVID-19, while 163 in the saline or control group became infected. This represents a 94% efficacy. Here are the curves comparing the placebo, the upper line, to the vaccinated, the lower line. The vertical axis is the number of cases as a percent infected, and the horizontal line, time in days. Within 12 to 14 days of receiving the first dose of the vaccine, you can see the number of cases flattens in the vaccine group, while cases continue to accumulate in the saline or placebo group. When it came to severe cases, there was 1 in the vaccine group and 9 in the placebo group. 2 indicators that predict protection are the level of antibody generated against the spike protein and the level of neutralizing antibodies. As shown on the left, the Moderna mRNA vaccine stimulated high immunoglobulin titers over time, titers rising to higher titers than natural infection, reaching levels of 300,000 to 3 million, shown in the left figure, and neutralizing antibodies are shown in the right figure. Mixing of the serum with virus kills the virus at dilutions of 1 to 300 to 1 to 400 in vaccinated subjects, but not in the placebo. As noted with the Pfizer vaccine, efficacy is over 90% when it comes to preventing mild to moderate disease, and 100% were protected from contracting serious disease. Recent studies among healthcare providers and nursing home residents have confirmed a greater than 90% efficacy under real-life conditions. Next are the viral vector vaccines. Both the Oxford, AstraZeneca, and the Johnson & Johnson vaccines, as well as the Sputnik V vaccine, have used an adenovirus that is unable to replicate or produce new virus. This single-stranded DNA virus has been used in gene therapy for over 20 years. The DNA message for the spike protein has been inserted into the virus, and when the virus is injected intramuscularly, it invades immune processing cells where it generates messenger RNA for the spike protein that then guides the synthesis of that protein. Once synthesized, the spike protein migrates to the cell surface where B and T cells generate antibodies and program cells to kill the SARS-CoV-2 virus. These vaccines have also completed phase 3 trials. An analysis of the Oxford AstraZeneca vaccine has been published in Lancet. The trial has 23,848 participants from the United Kingdom, Brazil, and South Africa, and requires 2 doses of vaccine. Control subjects received the meningococcal vaccine or saline. As you can see, the vaccine was protective. Compare the blue meningococcal vaccine recipients to those receiving the adenovirus, the orange line. These preliminary studies demonstrate the efficacy to be 71%. A third strategy is the tried and true approach to vaccines, injecting a surface protein to induce immune response. This approach has proved very effective for encapsulated bacterium Strep pneumoniae, sometimes called the pneumonia vaccine, commercial name Prevnar 13, and for Haemophilus influenza type B, abbreviated Hib vaccine, has nearly eliminated this once deadly infection in children. For the SARS-CoV-2 vaccine, the full-length spike protein is expressed and purified and combined with an adjuvant, matrix M1 adjuvant, a component that amplifies the immune reaction and is injected intramuscularly, and the immune processing cells take up the foreign protein and express it on their surface, stimulating B and T cell immune responses. Early studies published in December of 2020 reveal that this vaccine induces a robust antibody response to both 5 micrograms and 25 micrograms of protein combined with adjuvant, and the levels are comparable to infected patients who required hospitalization. See the values within the black box, achieving levels of antibody of 47,000 to 63,000 ELISA units per ml. This approach also induces impressive viral neutralizing titers of 1 to 4,000. The fourth strategy is to inject inactivated or killed whole virus. China has used this approach to produce the CoronaVac and Sinopharm vaccines, and India to produce Covaxin. Manufacturers in both countries have taken whole virus grown in viro cells and inactivated the virus with beta-propionolactone. The advantage of these vaccines is that they immunize to a broad range of epitopes or protein sites on the virus. Covaxin and Sinopharm vaccines have achieved an efficacy of 70 to 80% range, while CoronaVac has demonstrated lower efficacy of 50%. The fifth strategy being used is to create a live, attenuated, or weakened virus that has parts of its RNA deleted so it no longer can cause significant disease. The virus will be injected and is expected to induce an immune response without causing significant harm to those vaccinated. There is a limited data on the vaccine, which is being studied in India and has not yet been released. Here is a table summarizing the effectiveness of the current vaccines as determined by the Institute of Health Metrics and Evaluation, IHME. The most important assessment is the ability of each vaccine to prevent serious disease and hospitalization. The mRNA vaccines, Pfizer and Moderna, have the highest efficacies of 95 to 97% protection from severe disease caused by the ancestor strain, as well as the Alpha and Delta variants. Their efficacy for preventing serious disease caused by Omicron variants was lower, but in an acceptable range of 72 to 73%. With regards to efficacy for preventing infection, they were very effective, 84 to 94% for earlier strains. However, for the Omicron variant, their ability to prevent infection is low, 44 to 48%, and this has been true for all of the other vaccine classes. None of the vaccines can prevent infection. The next most effective are the DNA viral vector vaccines, AstraZeneca, J&J, and Sputnik V, with efficacy for preventing severe disease ranging from 89 to 94% for the earlier strains and 57 to 71% for the Omicron variants. The only FDA-approved vaccine, protein vaccine, Novavax, demonstrates comparable efficacies of 89 and 65%. For earlier SARS CoV-2 strains, the inactivated whole virus demonstrated somewhat lower efficacy in the mid-70% range, with the Chinese CoronaVac having the lowest efficacy of 50%. Protection against the Omicron variants is 37 to 57%. Overall, with the exception of the killed whole virus vaccines, the other vaccines offer reasonable protection against serious disease for all SARS-CoV-2 variants, including Omicron. To summarize the content of this video, the majority of the vaccines are directed against the full-length spike protein from the original human virus. The mRNA vaccines, Pfizer and Moderna, are highly effective at preventing serious disease, 95 to 97% efficacy for the earlier strains, and 72 to 73% for the Omicron variants. The adenovirus vector vaccines demonstrate nearly comparable protection, 89 to 94% for earlier strains and 57 to 71% efficacy for Omicron variants. The tried and true purified protein vaccines combined with an adjuvant have a similar efficacy to the adenovirus vaccines. Killed whole SARS-CoV-2 is somewhat less effective for protecting against disease, with an efficacy in the mid-70% range for earlier strains and 50% range for the Omicron variants. The Chinese vaccine CoronaVac is less effective, 50% for earlier strains, 37% for Omicron.